Hi, I'm Professor Amanda Kraus. I'm the Head of Division of Human Genetics here at the National Health Laboratory Service and the University of the Witwatersrand. We're a joint department, and in that are a multifaceted department with a service component, a research component, and a teaching and training component. As a service department, we have both a laboratory and a clinical service, with the advantage that we have a laboratory clinical interface and so see the patients, diagnose them and manage them in our genetic clinics. Our research is focused on rare genetic conditions and understanding those conditions in the context of a heterogeneous genetic background. We live on a continent where there is great diversity in Africa, and it is really important that we translate that into accurate patient management and diagnosis. We also have a very strong association with the Sydney Brenner Institute of Molecular Biosciences, whose focus is on bioinformatics and more complex disease. And we share a student body of masters and PhD students across the large research division. We also teach health sciences students at undergraduate level, and at postgraduate level have a number of teaching and training programs. We also train interns in laboratory science in the field of human genetics. We also offer postgraduate training programs in medical genetics. We have a registrar training program as well as a genetic counselling program.
I am the genetic counselling manager at the human genetics department of the NHLS and Wits, and what that means is that I work with the clinical team. So we've got a clinical team of about 20 people, which consists of medical geneticists, so doctors who've specialized in genetics, medical genetic registrars, genetic counselors, and genetic counseling students and interns. So what we do is we see patients who are at risk of having a genetic condition or have a genetic condition in the family and have been diagnosed with it. We see patients in a number of hospitals in and around Joburg, both in the state and the private sector. And what we do in those clinics is help to diagnose an underlying genetic condition, help to recommend the appropriate testing, feedback the results of the test, and then explain to a patient what it would mean for them and their family members if a genetic diagnosis is made. The counseling part of it is obviously the fact that we also help people come to terms with the psychosocial impact that having a genetic diagnosis would have on them, their loved ones, their families, and how they cope with that.
I am Lilobile Masehlele from the Cytogenetics Laboratory in the Division of Human Genetics. In our lab, we look at various types of cases which may arise due to numerical abnormalities, which are gains or losses of a chromosome, or structural abnormalities, which are chromosome structures that have been arranged as a result of a deletion, duplication, inversion, or a translocation, and so forth. The types of tests that we run in this section are karyotyping, which is a conventional test which entails the pairing and ordering of all chromosomes, thus providing a genome-wide picture of an individual chromosome. Macroarray, which is an array-based comparative genomic hybridization test that evaluates the areas of the human genome for gains and losses of chromosome segments at a higher resolution compared to karyotyping. Quantitative fluorescent polymerase chain reaction, QF-PCR, which is a rapid test that entails amplification, detection, and analysis of chromosome-specific DNA sequences known as small tandem repeats that are located on the chromosome of interest, and thus we can determine the common autosomal, trisomies, and sex chromosomes. The results of all these tests can either confirm or disprove a diagnosis.
I am Famida Esop, a medical scientist, and I head up the Molecular Genetics Diagnostics section. In our section, we look at a number of human genetic diseases which are caused by mutations in many different genes. The type of tests we run in this section include testing for mutations which cause these different genetic diseases, including common conditions such as albinism, cystic fibrosis, and Fragile X syndrome. The technology we use includes PCR, sequencing, and MRPA. The results from these tests can show the presence or absence of a mutation, in which case it will either confirm or exclude the suspected diagnosis. I also head up the Health Professions Council of South Africa Intern Medical Scientist Training program in our division. And this is a 24-month training program during which the intern will rotate through each of the different diagnostic sections. On completion of the training, the intern can register with the HPCSA as a medical biological scientist in genetics.
My name is Mariam Dao. I'm a senior medical scientist in the Division of Human Genetics. We utilize a technology called next generation sequencing to do our testing. Here we utilize inherited disease panel. This panel has 500 genes. The benefit of using this panel is that we can batch these patients in one panel and then analyze them differently. We have successfully integrated this technology into our diagnostic setting, and we are very happy about that because there's no other diagnostic lab that utilizes a panel like ours to test for monogenic disorders.
Hello, my name is Rajesh Mahabir and I'm a medical scientist in the DNA profiling lab at the Department of Human Genetics. The DNA profiling lab is responsible for paternity, maternity and kinship testing. These tests look at the relationships between individuals. For example, a paternity test looks at the relationship between an alleged father and his proposed child. When we look at the results from the test, we look at DNA markers between the alleged father and the child. The child needs to have 50% of the markers from the father. The results from the test can either support or negate the alleged relationship.
My name is Michelle Ramsey and I'm a professor of human genetics. I've been part of the division for the past 35 years, but in 2014 I moved to the Sydney Brenner Institute for Molecular Bioscience, where I'm currently the director. At the institute we have masters and PhD students and postdoctoral fellows, as well as many scientists. Our link with the Division of Human Genetics is that all our students who do genetics projects are doing this through the Division of Human Genetics and therefore join all the activities that all the students in human genetics would do. So our emphasis here is really looking at genetic diversity in African populations and trying to understand how they contribute to health and disease. Essentially, the skills that we have here are data analysis and particularly bioinformatics, as that is a very important part of what we do. So at the Sydney Brenner we have a biobank and we have a lot of data, and that data can be used for many different projects. So we have collaborations both within Wits but also on the continent and further afield. So there are wonderful opportunities here to do research.
Hi there, I'm Zanelombard. I'm the postgraduate research coordinator in the Division of Human Genetics. I want to tell you a bit more about the opportunities that we have for postgrad students and for research in the division. So we've got several different opportunities for postgraduate students that include our Bachelor of Health Science in honours, the MSc Med in Human Genetics, as well as PhD opportunities. And we also have a number of different supervisors, an array of different research interests that people can get involved in. So the research that we do in the department is mostly focused on looking at what role genetics plays in different human diseases. And these include things like neurological disorders like Huntington's disease, developmental disorders in children, cancers in children, as well as ovarian and breast cancer in adults, as well as inherited skin disorders, things like albinism and vitiligo, just to name a few. So to mention, from 2022 we will also be offering a new field of study in our MSc Med course, which is called Genomic Medicine. This is going to be a full-time one-year master's degree by coursework, where there will be examinable coursework components as well as a small 30% research component that students can enroll for and participate in. What you can do is reach out to me to discuss any of the opportunities that we have and potential supervisors that are available, and we can match you up and make sure that you get your application in to join the division and complete your research here.
I am passionate about ensuring that patients who require genetic services get appropriate service and appropriate management. In order to do that, we have to be able to translate critical basic research and understanding so that patients get appropriate care.